In this video we will discuss about L-selectin and its structure. This L-selectin, basically its role is to modulate cell adhesion, migration and cell signaling, and we have seen some of its role in the previous video where we have discussed how this it helps this dendritic cells or the immune cells to reach to the tumor sites. So by reducing the circulation speed and through this L-selectin interactions. So in this video we will discuss the structure about the L-selectin. So L-selectin basically is a type 1 glycoprotein. So type 1 glycoprotein means its N-terminal is on the extracellular side and C-terminal is on the cytoplasmic tail. And in the N-terminal side or in the extracellular domain, it has different domains. In the extracellular sites it has different domains. For example, this C-type lectin domain, it is basically a calcium dependent lectin domain, and thereafter this epidermal growth factor like domain or EGFR domain. We have seen this more common when we discussed about the drugs that target the cancers. Some of them usually target this EGFR, which is overexpressed in the cancer cells. Thereafter this sequence consensus repeat domain and this cleavage site. This is to cleave off the cellular domain, thereby preventing the L-selectin's role to trigger intercellular functions. In the intercellular part, it triggers different functions, and the region of amino acid is highlighted to show that which amino acid sequence plays a role in which functions. This CA, which is the calcium mediated binding, it is basically triggered by this KK amino acids or lysine amino acids, and it usually prevents the shedding of this L-selectins. After that, this ERM is basically control between this L2K amino acid sequence, and its role is to cause this shedding and microvillar localizations. So both of them are have a different functions. And this alpha actinin or rolling, whereby reducing the speed of the cells, usually triggered by this amino acid sequence, which are highlighted in this blue color. And they also compared these L-selectin structures with the mouse L-selectins. They are able to find out that there is a variant that in the mouse lectin there is only one serine present at 364 position, whereas in the case of human L-selectin, there is another serine present at the 366 position. So this is the difference, one of the difference between L-selectin that is found in the human and the L-selectin that is found in the mice. Apart from that, there are two different variant splice variant are found in the mouse L-selectin, and the region and the sequence that is unique to each variant is highlighted, is underlined, that is shown here, that is QEISRKDG. This amino acid sequence is specific to the variant 1, and the CAL PHL STF1 is specific to the variant 2. So these are some of the difference between mouse L-selectin and human L-selectin, and this needs to be taken care in mind while designing the in vivo studies. And also this cleavage site in the human, the cleavage site is shown here, and we can see that when they compare the human cleavage site and the mouse cleavage site, a region of the amino acid sequence are conserved. However, the upstream of the cleavage site sequence are differ. That can be seen here, that in the human L-selectin, the cleavage site contains KLDKS. In the mouse L-selectin, it contains ETNRS. So this indicates that cleavage sites are also differ based on the species. And one thing that I forgot to tell you is this L-selectin, since it is a type 1 glycoprotein, its size also varies based on the cells where it expresses, and the region is that based on the cells, the glycosylation expressions on this L-selectin varies. So, for example, in the case of lymphocytes, its size is around 65 kilodalton, which has a less glycosylated L-selectin, whereas in the case of neutrophil, its size is around 100 kilodalton, which has more glycosylated L-selectin, or they will have a more glycosylations, more glycans that are present on the L-selectin. So these are some of the structural and the structure. These are some of the salient aspect of this L-selectin in terms of the different domains and their size and their how they differ between mouse and human being. If you need to understand more about this L-selectin and its function, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.